Welcome to Reads Biology. Today we will discuss question answers of plant kingdom. So, let's start. The first question: what is the basis of classification of algae? The answer will be Fritz in 1935 has classified algae considering phylogeny, affinities, and interrelationships of various forms. He classified algae mainly on the basis of characters like structure of plant body, nature of the pigments, reserve food material, number of position of flagella, chemistry of cell wall, and methods of reproduction. Algae is divided into 11 classes, but among them, three main classes are chlorophyceae, phaeophyceae, and rhodophyceae. The next question: when and where does reduction division takes place in the life cycle of a liverwort, a moss, a fern, a gymnosperm, and an angiosperm? So let's see. All of these plants show life cycle with one gametophytic end generation and one sporophytic to end generation. Reduction division or meiosis that produces haploid N cells from diploid cells, which is 2N, is necessary in their life cycles to restore gametophytic generation after sporophytic generation. To occur, it occurs in different body structures according to the basic body design of these groups. And reduction division in a liverwort and moss takes place at the end of sporophytic generation, where haploid spores are formed by reduction division of spore mother cell inside the capsule. Spores germinate to produce dominant gametophytic generation. Reduction division in fern takes place at the end of dominant sporophytic generation inside the sporangium from spore mother cell by reduction division or meiosis. Spores may be of one type, which is homospory, or of two types, heterospory. Reduction division in gymnosperms takes place at the end of dominant sporophyte generation. Megaspore and microspores are produced by the reduction division of diploid megaspore mother cell and diploid microspore mother cell respectively inside megasporangium and microsporangium. Reduction division in angiosperms takes place at the end of dominant sporophytic generation. The haploid pollen grain or microspore and the haploid egg cell are produced by reduction division of diploid microspore mother cell and diploid megaspore mother cell respectively. Microsporic division occurs inside anther and megasporic division occurs inside gynoecium or ovary. The next question: name three groups of plants that bear archegonia. Briefly describe their life cycle of any one of them. So let's see. The three groups of plants that bear archegonia are bryophytes, pteridophytes, and gymnosperms. Life cycle of a bryophyte is as follows. The main plant body of bryophyte is gametophytic or haploid or N, which is independent and may be thallose. No differentiation in root, stem, and leaves. and Riccia is an example of this. Or it may be foliose, having leafy axis, and Funaria is an example of this type. The dominant phase in the life cycle of Funaria is the gametophyte, which occurs two stages, the protonemal stage and the erect, leafy gametophytic plant. The leafy gametophyte consists of an upright, slender axis, which is stem-like, and that bears spirally arranged leaves. It is attached to the substratum by multicellular branched rhizoids, which is a root-like structure. Vegetative reproduction takes place by fragmentation, where the buds formed in secondary protonema. The sex organs, antheridia and archegonia, are produced in dusters at the apices of the leafy shoots. Antheridia produces antherozoids and archegonia produces egg. Antherozoid, which is male gamete, and egg, female gamete, fuses and form zygote. Zygote develops into sporophyte, which is differentiated into foot, seta, and capsule, and spores are produced in the capsule. Spores, on reaching a suitable substratum, germinate to produce a filamentous juvenile stage called the primary protonema, which later produces secondary protonema that forms erect leafy plants. The next question: mention the ploidy of the following: protonemal cell of a moss, primary endosperm nucleus in dicot, leaf cell of a moss, prothallus cell of a fern, gemma cell in Marchantia, meristem cell of monocot, ovum of a liverwort, and zygote of a fern. So let's see one by one. Protonemal cell of a moss is haploid. Primary endosperm nucleus in dicot is triploid because it occurs by double fertilization. Leaf cell of a moss is haploid. Prothallus cell of a fern is also haploid. Gemma cell in Marchantia is also haploid. And meristem cell of monocot is diploid. Ovum of a liverwort is haploid. And zygote of a fern is diploid. Write a note on write a note on economic importance of algae and gymnosperms. So let's see one by one. The economic importance of algae is as follows. The group of algae plays a role both economically as well as it has harmful roles. The beneficial importance of algae are: people of coastal countries have been using seaweeds and certain other algae as source of food like porphyra, alva, laminaria, etc. Some algae are used as food for marine as well as domestic animals like sargassum, macrocyst, etc. Algae are useful source of many commercial products like agar, a jelly-like substance which is a complex polysaccharide, and it is extracted from a species of red algae belonging to the genera Gelidium, Gracilaria, etc. Agar is used as a base in culture media in microbiology lab to culture bacteria. Carrageenan occurs as a whale cell wall polysaccharide and which is esterified with sulfate. It is extracted from red algae like Chondrus crispin etc. And it is used in pharmaceutical emulsifier and textile, leather, cosmetic industries. Alginates are salts of alginic acid found in the cell wall of Phaeophyceae, which is brown algae like fucus, laminaria, etc. Algae are also useful in medicine industry. Antibiotic chlorelin is obtained from chlorella. Extracts of cladophora, lingbia kill strains of Pseudomonas and Mycobacterium like bacteria. Nutella is used to destroy mosquitoes' growth in ponds and used to control the malaria. Some algae are used in agriculture like nostoc and abina are used to convert atmospheric nitrogen into nitrogenous compound which are absorbed by higher plants. Some seaweeds like fucus, lycophyllum, etc. are rich to potassium, phosphorus, and these are trace elements and growth substances and are used as fertilizers by coastal people. Some algae like chlorella, chlamydomonas, etc. are used in sewage disposal in ponds. These algae help in bacterial decomposition by providing oxygen. Some algae like chlorella, cyanococcus, etc. are used in space travels. A person inside a spaceship will need a device to get rid of carbon dioxide and other body waste and will require sources of oxygen and food. These algae are very useful for this purpose. A large amount of iodine, a mineral element present in thyroxine hormone of thyroid gland, is extracted from kelps, which is brown seaweed, or members of Phaeophyceae or brown algae like laminaria, fucus, ascophyllum, etc. Similarly, red algae like rhodomela, polysiphonia, rhodymenia are sources of bromine. Then we will discuss the harmful importance of algae. Some blue, green, and green algae like Rhococcus, oscillatoria grow over the surface of water bodies in abundance and cause the water to bloom on death and decay of these algae give a bad smell. Some algae secrete poisonous or toxic substances and kill other aquatic animals and plants. Parasitic algae like Cephalurus virescens causes red rust in tea, coffee plant, etc. Economic importance of gymnosperms. Some species of cycas like Cycas revoluta, Cycas rumphi look like palm tree and are used as decoration purpose as they remain fresh for a long time. Stem portion of Cycas revoluta is a good source of sago, which is a kind of starch used in making bread by poor people. Seeds of some species of Cycas are roasted and taken as food. Young succulent leaves of some species of Cycas are cooked as vegetable. Many gymnosperm have medicinal value. The fresh juice extracted from the Cycas sarcinalis leaves is used as medicine for stomach disorders, blood vomiting, and other skin diseases. Pollen grains of some cycas plants are reported to have some narcotic effect. Some gymnosperms like pinus, abies, citrus are the chief source of various types of woods. The wood of juniperus is used in making pencils, scales, holder, etc. Some species of pinus is a good source of turpentine, wood gas, wood alcohol. The next question. Both gymnosperms and angiosperms bear seeds. Then why are they classified separately? So this is a very important question and the answer will be gymnosperms and angiosperms both bear seeds. But they are classified separately because gymnosperms are a group of plants in which the ovules are freely exposed on open megasporophytes, whereas in angiosperms the seeds or ovules are enclosed within ovary which later forms the fruit. So this is the main difference between gymnosperms and angiosperms. Next question. What is heterospory? Briefly comment on the significance. Give two examples. So let's start. The occurrence of two kinds of spores in same plant is called heterospory. Among them, the smaller spore is called microspore and the larger spore is megaspore. Micro means small and mega means big. Heterospory first evolved in pteridophytes. Significance of heterospory. Heterospory is associated with the sexual differentiation of gametophyte. A microspore develops into a male gametophyte, whereas a megaspore develops into a female gametophyte. In homosporous pteridophytes, spores have to germinate on soil, thus face more environmental problems. In heterosporous pteridophytes, spores germinate within the sporangium and the gametophytes are retained inside for variable periods of time. Hence, germinating gametophytes has better chances of survival. This lays the foundation of complete retention of gametophytes within sporophytes in angiosperms and gymnosperms. Heterospory is the basis of development of seed habit in higher plants. Next question. Explain briefly the following terms with suitable example. Protonema, antheridium, archegonium, diplontic, sporophyll, and isogamy. Let's see one by one. First, protonema. It is the first, usually branched, green, and filamentous structure produced by a germinating moss or fern spore. The protonema of mosses bears buds that develop into the gametophyte plant. In fern, the protonema becomes the prothallus. Antheridium. It is the male sex organ of cryptogams. Cryptogams means algae, fungi, bryophytes, and pteridophytes, and it is known as antheridium, the male sex organ of these plants. It produces the male gametes or antherozoids. It may consist of a single cell or it may have a wall that is made up of one or several layers forming a sterile jacket around the developing gamete. Archegonium, the multicellular, flask-shaped female sex organ of bryophytes, pteridophytes, and many gymnosperms is known as archegonium. Its dilated base called the venter contains the female gamete or egg or oosphere. The cells of the narrow neck of archegonium liquefy to allow the male gamete to swim towards the oosphere. Diplontic. It is the kind of life cycle in which the diploid sporophyte is dominant and this diploid phase is photosynthetic. The gametophytic phase is represented either by gametes only that are formed through meiosis or by a highly reduced few-celled gametophyte. Example, all seed-bearing plants like gymnosperms and angiosperms. Sporophyll. It is a type of leaf-bearing sporangia. In ferns, the sporophylls are the normal foliage leaves, but in other plants, the sporophylls are modified and arise in specialized structures such as strobili or of club moss, gymnosperms, and other flower of angiosperms. In most plants, the sporophylls are of two types, microsporophylls and megasporophylls. Isogamy. It is a type of sexual reproduction where fusion takes place between two identical gametes. The gametes are similar in size and structure and they show equal motility during sexual reproduction. Example Spirogyra. It is an algae. Differentiate between the following red algae and brown algae. Always write difference in this pattern. Make three columns. First column feature, second column first difference, and third column second difference. So our first feature is major pigment. Major pigment in red algae is chlorophyll and phycoerythrin, so it is red. And major pigment for brown algae are chlorophyll and fucoxanthin, that is why it is brown in color. Second feature stored food material, fluoride starch for red algae and mannitol and lamellin for brown algae. Third feature cell wall composition, cellulose for red algae, for brown algae, cellulose and algin. Fourth feature presence of flagella, absent for red algae and present for brown algae, and the number of flagella is 2, and they are unequal in size and lateral in position. Fifth feature structure of thylakoid. Thylakoids are unstacked for red algae, and thylakoids occur in group of 3 for brown algae. Next difference homosporous and heterosporous pteridophytes. Again make 3 column. First feature types of spores. Only one kind of spore is produced for homosporous pteridophytes. Homo means same and hetero means different. For heterosporous pteridophytes, two kinds of spores are produced. Second feature size of spores. Spores are equal in size for homosporous pteridophytes and different in size the size of spores for heterosporous pteridophytes. Smaller spores are microspores and larger spores are megaspores because micro means small and mega means large. Third feature source of spores. Spores are produced from same sporangia for homosporous pteridophytes. Microspores are produced from microsporangia and megaspores are produced from megasporangia for heterosporous pteridophytes. Fourth feature spores develop. For homosporous pteridophytes, spores develop one kind of gametophyte. And for the second type, heterosporous pteridophytes, microspore develop into male gametophyte and megaspore develop into female gametophyte. Fifth feature place of germination. Spores germinate in soil and produce independent gametophyte for homosporous type, and for heterosporous, spores germinate within sporangia and produce dependent gametophyte. Then sixth feature example Lycopodium for homosporous pteridophytes and Selaginella salvinia for heterosporous pteridophytes. Let's see the second. Next question differences between liverworts and mosses. Again, make 3 columns. First feature structure of gametophytic plant body. For liverworts, gametophytic plant body may be thallose, Riccia, or foliose. For mosses, gametophytic plant body differentiated into prostrate, branched, filamentous, thalloid, protonema, and leafy erect gametophore. Second point, types of rhizoids present. Rhizode is a root-like structure. For liverworts, on the ventral surface of thallus, unicellular rhizoids and simple multicellular scales are present. For mosses, rhizoids are multicellular, branched with oblique septa. Third point, origin of sex organs. Sex organs are present on dorsal surface of thallus and develop from superficial cells for liverworts. For mosses, sex organ developed from superficial cells at the apex of leafy gametophyte. Fourth point, presence of elaters, generally present but absent in Riccia, and for mosses it is absent. Presence of stomata, absent in the wall of capsule for liverworts, and for mosses present in sporophyte for gaseous exchange. Dehiscence, for liverworts, dehiscence of capsule is irregular, and for mosses, dehiscence is regular. The next question, differences between syngamy and triple fusion. This is very important question. So let's see the answer. Again make three columns. The first feature, types of fertilization. For syngamy, it is the generative fertilization, and for triple fusion, it is vegetative fertilization. Second feature, involvement of gametes. For syngamy, both male and female gametes are involved. For triple fusion, only male gamete and two vegetative nuclei are involved. Because it is triple fusion, it is done by double fertilization. Third feature, product. Syngamy produces a diploid zygote and triple fusion produces a triploid primary endosperm cell. Fourth feature, end product. Zygote forms embryo for syngamy and primary endosperm cell produces a food-laden endosperm. How would you distinguish monocots from dicots? And this is a very important question. So let's see the differences. Again make three columns. The first feature, number of cotyledons. For monocots, seed contain only one cotyledon. Mono means one and di means two. For dicot, seed contain two cotyledons. Second feature, structure of flower. Flowers are trimerous. Tri means three. For dicots, flowers are pentamerous. Penta means five or tetramerous. Tetra means four. Third feature, number of germ pores in pollen grain. Pollen grains generally possess a single germ pore for monocots. For dicots, pollen grains commonly have three germ pores. Fourth feature, structure of leaves. Leaf possess parallel venation. For dicots, leaf possess reticulate venation. Fifth feature, types of roots present. For monocots, primary root is short-lived, taproot is absent, and adventitious root are present. For dicots, primary root is long-lived and it forms taproot. Adventitious root are found in some cases, but not all the time. Sixth feature, structure of stem. In stems, ground tissue is not differentiated into cortex and pith. For dicots, in stem, ground tissue is differentiated into cortex, pith, and endodermis. Seventh feature, structure of vascular bundles. Vascular bundles are scattered and closed for monocots. For dicots, vascular bundles are arranged in a ring and it is open. Eighth feature, Presence of cambium. Cambium is absent for monocots and cambium present for dicot. Ninth feature. Presence of pith in root. In root, a pith is always present for monocots and for dicots, in root, pith is absent. Tenth feature. Number of vascular bundles. Vascular bundles are many, more than 8 for monocots, and for dicots, vascular bundles are few. 11th feature. Structure of vessels. Vessels are rounded in outline for monocots, and for dicots, vessels are polygonal in outline. So these are the 11 points. The difference between monocots and dicots. So let's see the next question. Match the following, column 1 to column 2. So let's match. First, Chlamydomonas. It will match with algae because Chlamydomonas is an algae. The next B cycas will match with gymnosperm because it is a gymnosperm. Next one, Selaginella is a pteridophyte and sphagnum is a moss. So this is very easy. So let's move to next question. Describe the important characteristic of gymnosperms. So let's see the answer. The term gymnosperm is derived from two Greek words. Gymnos means naked and sperma means seed. Naked seeded plant is gymnosperm. So gymnosperms are a group of plants in which the ovules are freely exposed on open megasporophytes. The important characteristics of gymnosperms are living gymnosperms are perennial and vary from predominantly medium-sized trees like cycas to tall trees like pinus and shrubs like ephedra. Plants possess taproot system. Some genera possess symbiotic relationship of nitrogen-fixing algae in coralloid roots like cycas and fungi in mycorrhizal roots found in pinus. The stems are aerial, erect, branched or unbranched in case of cycas and woody. The leaves may be simple or compound. They are scaly and foliage also. Leaves are well adapted to withstand extreme of temperature, humidity, and wind. Roots are characterized by the presence of diarch to polyarch vascular bundles, and the xylem is exarch. Stems are provided with collateral, endarch, and open vascular bundles, which are arranged in a ring. Secondary growth is present and annual rings are formed. Xylem contains xylem parenchyma and tracheids with bordered braids and vessels are absent, except natum, ephedra, and Riccia. Phloem contains sieve cells and phloem parenchyma, and companion cells are absent, except in natum, ephedra, and Welwitschia. These are exceptions. Leaves are protected by thick layers of cuticle. Sunken stomata are present. Mesoxylem and transfusion tissues are found in the leaves. Palisade tissues and spongy parenchyma may be present in mesophyll or it may be undifferentiated. Reproductive organs form cones or strobilus except female organs of cycas. Male cone is made of overlapping microsporophylls that bear microsporangia on abaxial side which produce microspores. Female cone is formed by overlapping megasporophylls which bear ovules or megasporangia. Ovule is orthotropous Antimic, unitegmic, with three layers: outer fleshy, middle stony, and inner fleshy layer. Nucleus of ovule contains single megaspore mother cell, which undergoes reduction division or meiosis to form four megaspores, out of which three degenerate and only one survives. So gymnosperm is heterosporous, which produce megaspores and microspores. Single megaspore forms haploid, female gametophyte, or endosperm before fertilization. At micropylar end of female gametophyte, two or more archegonia are produced. Archegonium is with reduced neck, with no neck canal cell. Microspores are released from microsporangium and are carried in air currents and come in contact with the micropyle of the ovule. Pollen tube carrying the male gametes grows towards archegonia and discharges its content near the mouth of the archegonia. After fertilization of zygote, zygote forms or spores give rise to embryo proper and the ovules develop into seeds. Polyembryony, which is development of more than one embryo, is an usual feature of gymnosperms, but only one of them survives at later stage. In embryo, two or many cotyledons are present. The seeds of gymnosperms are uncovered. So, thank you so much for watching. Like, share, and subscribe to Reads Biology. Next day, we will start Chapter 4, The Animal Kingdom. Take care, everyone. Have a nice day.